Okay, this is part 2 of the genetics chapter 9. Hopefully you've gotten through the DNA replication portion of this chapter. This portion is going to concern itself with transcription and translation, or protein synthesis, how we take the information from DNA and put it into the form of a protein. When we're talking about DNA and DNA replication and the whole process of DNA replication and getting everything out of that code that we have to remember that DNA is just a master code. There's nothing we can do with it until we transcribe it and translate it. There is something called the central dogma, which basically states that the flow of genetic information goes from DNA to RNA to protein. So DNA is our master code. We're going to use that as a template to synthesize an RNA molecule. So from going from DNA to RNA, this is called transcription. Once we have an RNA molecule, it is still a coded process, so we can't see any expression of a trait until we take that RNA molecule and produce a protein. So once we take our RNA and make a protein, then that is translation. You do have an exception to this central dogma rule with RNA virus and retroviruses. They are going to have to make an RNA molecule. That RNA molecule, their RNA genome, is going to have to be transferred to a DNA molecule. Then they will go from DNA to RNA to protein. But in every living system, you will have that central dogma will be followed: DNA to RNA to protein. If you look here, you'll see this picture. We've got our DNA molecule. We can replicate that DNA molecule, or we can go from DNA to making a messenger RNA or our accessory RNA molecules to translation. So when we go DNA through transcription of DNA to messenger RNA then will be translated into a protein, and the expression of that DNA then will be our trait. DNA is going to be represented in a triplet code, or 3 consecutive bases on a strand of DNA. A gene differs from one gene from another in the composition of those triplet codes. So the sequence of my triplets is going to give me my gene. Each DNA triplet is going to act as a template to produce something in RNA called a codon. This is a specific sequence of 3 nucleotides in messenger RNA, and these are going to make up our codon. We're going to talk a lot about codons here after a bit when we talk about translation. Each codon is going to represent a code for a particular amino acid. When the triplet code is transcribed and translated, it's going to dictate a particular order of amino acids in a particular protein chain. And that order of amino acids is going to ultimately determine the sequence of that protein, and sequence determines shape, and as we know, shape determines function. Proteins will ultimately determine phenotype, or the trait. Either the protein itself will be the trait, or it will establish a trait by causing some other mechanism to be turned on or turned off. DNA is mainly the blueprint. It's going to tell us what we need, what kinds of proteins will be made, what kinds of RNA will be made, and how to make them. It is just the code. It doesn't do any more or any less. Here's a great picture that's from your book. It's actually on page, let me double check here. It's on page 241. And if you look here, this top row is going to represent our DNA, and we're going to take one strand of this, and on this picture it's using the very top strand, and you'll see that the DNA is clustered into these triplets. This template here, this strand of DNA is going to act as a template to give us a strand of messenger RNA, and this messenger RNA is a complement to our DNA. This strand of messenger RNA, these triplet nucleotides are our codons, and each sequence here, each codon sequence, is going to code for a particular amino acid. Here we have 5 different codons, so we will have 5 different amino acids. When you look at the number of nucleotide bases, if that nucleotide base sequence is divisible by 3, you can tell how many amino acids you have. Here we have 15 single nucleotides, and if we take 15 and divide it by 3, we'll see that we have 5 amino acids. So if we have a sequence of DNA, we can know how many amino acids we will have in the end of that sequence. When we talk about transcription and translation, this is essentially the decoding of DNA into a protein. There are a number of different participants in transcription and translation, but the most prominent ones are messenger RNA, tRNA. You have some regulatory RNA molecules, the ribosomes, a plethora of enzymes, as well as our raw materials of amino acids and the materials that make our amino acids. The major ones that I want you to focus on are messenger RNA, tRNA, ribosomal RNA, and the ribosomes themselves. The rest of them we'll just allude to, and whatever I have in my notes is what we will cover. Messenger RNA is the intermediate molecule between DNA and protein. It is a transcript of the structural genes of DNA. It is synthesized a lot like DNA replication. We're going to have a polymerase enzyme that's going to use DNA as its template, and that DNA then is going to, that polymerase enzyme will read that DNA template and synthesize a messenger RNA. The message of this, the message is going to be transcribed in this mRNA, and it will be read later in translation as an amino acid sequence that we will use to make a protein. When we look at transcription, you have the enzyme RNA polymerase is going to be the enzyme that is the major enzyme of translation. It is going to be the enzyme that reads the DNA template and makes the messenger RNA. It's going to give you a complementary strand, just like in DNA, wherever you have one nitrogenous base, it will have its complement on the other strand. And we'll talk about the differences in the complementary sequences from between DNA and RNA here in a second. When we're going through transcription, prokaryotic cells and eukaryotic cells take on transcription in different parts of the cell. In the prokaryotic cell, transcription takes place in the cytoplasm, and in eukaryotic cells, it takes place in the mitochondria. This is going to have an effect on the differences between eukaryotic and prokaryotic transcription and translation, and we'll talk about that towards the end of this lecture. Messenger RNA is going to be an intermediate molecule between the storage of information in DNA and the translation of proteins at the end of the process. RNA polymerase is going to bind to a region on DNA called the promoter. It recognizes a very specific sequence in DNA, and that promoter region is going to act as a guide to synthesize messenger RNA. And this DNA then is going to act as a template. So once RNA polymerase finds that promoter region, it will read the DNA and give us our messenger RNA. Nucleotides in RNA are the same as those in DNA with one exception. Instead of having thymine like we have in DNA, we will have uracil in RNA. Binding is going to be adenine bonds to uracil and cytosine bonds to guanine. So the only thing that changes is that adenine doesn't bind to thymine anymore because we have no thymine in RNA. Everything else stays the same. So if I have an adenine in my DNA template, my complement to that in my messenger RNA will be uracil, not thymine. RNA polymerase is going to continue down a strand of DNA until it reaches a site on the DNA called the terminator. At the terminator, RNA polymerase recognizes that sequence, and it will then come off of the DNA template, release the messenger RNA strand, and then that RNA polymerase will either go back to the beginning, to the promoter sequence, and give us a new strand of messenger RNA, or it will be destroyed. But it just simply goes and reads the template and gives us a messenger RNA, and the sequence is going to go from promoter to terminator, and it's going to sequence, or it's going to copy everything in between into an mRNA molecule. Once we have transcription taken care of, we still have a coded message, so we still have to translate it. So translation is going to be decoding the information held in that messenger RNA into a functional, usable protein. There are two more RNA molecules that are going to become involved in translation. The first one is ribosomal RNA, or rRNA, and transfer RNA, or tRNA. Transfer RNA probably has one of the bigger jobs in translation. It is going to be essentially the key. The molecule itself even looks like a key. It contains a sequence of bases that are going to form hydrogen bonds to each other, and these are going to allow this molecule to bend back on itself and make a number of hairpin loops and give tRNA a clover leaf type of a structure. It gives us kind of a three-dimensional helix. If you look here, you'll see all of these sequences here are held together by hydrogen bonds. This clover leaf gives the tRNA a very specific structure. If you look at the bottom of the loop, you have an exposed set of nucleotides right here, and this is called the anticodon. This is going to be complementary to the messenger RNA on the codon on the messenger RNA. That anticodon is going to be like the other side of the Velcro. So when tRNA is brought up to meet that messenger RNA, the codon on the messenger RNA will bind to the anticodon on the tRNA, and it keeps that amino acid sequence complete and correct. Otherwise, you could have some slippage, and we don't want that. You'll notice at the opposite end of our tRNA, we have another sequence, and this is the amino acid binding site. This is where the amino acid binds to the tRNA and will then bind to, will come in contact with other amino acids and form peptide bonds. There are 20 amino acids, and for every amino acid, there is at least one specialized tRNA to carry it. Some of them, there are more than one, and we'll see what that code is all about. rRNA, or ribosomal RNA, is basically a structural RNA molecule. Its job is to compose the RNA, or excuse me, compose the ribosome. The prokaryotic ribosome, and as well as the eukaryotic ribosome, are composed of RNA and protein molecules tightly packed together. It's the interaction of the proteins and the rRNA that create the subunits of the ribosome, and this ribosome now has the ability to bind around the messenger RNA, read it, and translate it into an amino acid sequence. Here we have, if we're talking about a prokaryotic ribosome, we have our 50S ribosome, we have our 30S ribosome, and here is our strand of messenger RNA. Each of these, this strand of messenger RNA is going to be read in the ribosome. The ribosome is the translation factory, if you will. You're going to have your messenger RNA is going to be read in the ribosome, and each of these tRNA molecules will come and bind, let me go back, and will bind to a specific region on the messenger RNA, and you will see that these little green shapes here are to represent amino acids. Those will bind together, and the ribosome will continue to move down the sequence of messenger RNA until it reaches the end. We'll talk about those specifics here in just a moment. All of the elements that are needed to synthesize a protein are brought together into the ribosome to have this big, huge protein party. There are 5 stages of protein synthesis, or of translation. I'm going to give you these, but I'm just going to kind of talk about the whole sequence in one big sequence. I just want you to understand what the 5 stages are, and then we will go through and talk about mainly initiation, elongation, and termination. We're not going to talk a lot about protein folding and protein processing because those are extremely different in eukaryotic and prokaryotic cells. Just understand that after termination of translation, the protein is not a functional protein, but has to go through some protein folding to get that tertiary functional shape, and then some extra processing at the end. So our 5 stages are initiation, elongation, termination, protein folding, and protein processing. If we're talking about the initiation of translation, messenger RNA molecule is going to be made in the nucleus of the DNA if we're talking about a eukaryotic cell. DNA is going to be transcribed, and we're going to have our messenger RNA leaving, being transported out of the nucleus into the cytoplasm to the ribosome, where active translation will take place. The ribosomal subunits are specifically adapted to assemble and form around the messenger RNA and read it and bring the appropriate tRNA molecules into the sequence. Just as a reminder, prokaryotic ribosomes are different sizes than eukaryotic ribosomes. The prokaryotic ribosome is a 70S ribosome with a 50 and 30S subunits, and the eukaryotic ribosome is an 80S with 60 and 40S subunits. When translation is initiated, the smaller subunit is going to bind to the 5' end of the messenger RNA, just like we had DNA polymerase binding to the 3' end. When we are at the end of the whole process, the smaller subunit is going to recognize the 5' end of the messenger RNA and is going to bind to it. That larger subunit is going to come in and bind to the smaller subunit. Within that larger unit, you have all of the enzymes that are needed for making peptide bonds and binding these amino acids together. And you're going to see that all of that happens in sites within the large subunit. The ribosome is going to scan along the messenger RNA, and it's going to move from the 5' end to the 3' end. It's going to come to the very first codon that is called the start codon. The start codon in most proteins is usually going to be a codon sequence of AUG. This codes for the amino acid methionine. Prokaryotic cells have a specific start methionine, and eukaryotic cells have a specific start methionine. They aren't quite the same, but they still have the start codon of AUG. When that messenger RNA is in place, the ribosome will begin to bring tRNA molecules into spots on the ribosome called the P site and the A site. These P sites and A sites are going to guide that large subunit down the messenger RNA, bringing in the appropriate amino acid carrying tRNAs so the protein sequence can be established. There is a third site called the E site, and this E site is like the exit site, if you will. This is where used tRNAs that have given up their amino acids move into that E site to leave the ribosome so that they can go and be recharged, pick up a new amino acid, and come back into the sequence when they're called for. When you have the ribosome bound to the messenger RNA at that start codon, the first tRNA binds to the start codon, and in the P site, you're going to have that first tRNA bound. The anticodon is going to bind to the codon in the messenger RNA. The next tRNA in line is going to come and bind to the A site. When it binds to the A site, the amino acid in the P site is going to form a peptide bond with the amino acid in the A site. Once that bond is formed, the ribosome moves down, and what was once in the P site is now in the A site. What was once in the P site is now in the E site, and the ribosome is going to continue to move down. After the peptide bond is formed, you're going to have that ribosome move down the mRNA from codon to codon to codon. The used tRNA that was in the P site is going to move to the E site, and it's going to leave the ribosome to be recharged again. Okay? Then it's going to have the tRNA that was in the A site is now going to move to the P site. A new amino acid carrying tRNA now will move into the empty A site, and all of that process will happen again. The amino acid that is in the P site and the amino acid that are in the A site will form peptide bonds. The amino acid will leave the tRNA in the P site, and now we have that tRNA in the P site is empty. Ribosome moves down. The tRNA that was once in the P site that is empty is now in the E site. The tRNA that was in the A site that now has the new string of amino acids is now in the P site, and a new amino acid is going to come in and bond to that A site, and we're going to continue to do this until protein synthesis is complete. Translation is going to be terminated only when the ribosome comes to a specific sequence called a stop or nonsense codon. At this point, the ribosome recognizes that this is a non-polypeptide, a non-amino acid carrying spot, and it will separate then from the polypeptide chain. It will separate from the messenger RNA, it will release the polypeptide chain, and it is now free to go and bind to the start codon and translate this whole messenger RNA all over again. As the ribosome moves down the messenger RNA in prokaryotic cells, the start codon is exposed. And once that start codon is exposed, a new ribosome can bind and it will attach and begin another process. If you look here in this picture, you will see that you have got several different ribosomes on this one strand of messenger RNA, and that each of these polypeptide chains is in a different stage of completion. So as long as there is a start codon that is open and ribosomes that are available to bind, it will continue, and this is what you call a polyribosome. You have a number of different ribosomes on a single transcript. When amino acids are being coded for by these codons, there is something called a redundancy of the genetic code, meaning that a particular amino acid can be coded for by more than one codon. There are 64 codons that make up the alphabet of proteins. If you think of it this way, for every word that we have an alphabet of 26 letters. Every word in the English language, and there are multitudes of them, more than we could ever count, can be written with a sequence of those 26 letters. We can repeat letters, we can use letters singly, but we can write any word that we want by using those 26 letters. We can make any protein we want by using these sequences of codons in this redundant code. There are 64 codons. 61 of them are sense codons, or codons that code for a particular amino acid. The remaining 3 are nonsense codons, and these are going to be those termination sequences. There is what we call some wobble in the genetic code. Basically, it states that in many cases, only the first 2 nucleotides are required in the codon. That third one, you can have some variation or some mutation without altering the message. So sometimes these amino acids can have a little bit of give or a little bit of play, and you continue to keep the amino acid sequence the same and everything, then you have a functional protein, even though you may have a bit of a problem or variation or mutation in your amino acid, or in your codon at that third space. Here is basically the chart, if you will, on all of the amino acids. This is on page 248, or 246 in your book. What you do is you start with your first position. Let's say I have an amino acid sequence that's GAC. So I'm going to take my first base, which is G, my second base, which is A, and then my third base, which is C. So my GAC is aspartic acid, or aspartic acid, pardon me. So if you just follow first base, second base, and then the letter of the third base, you will be able to see that there are numerous amino acids here that have a multitude of different codons for them. Methionine is the only one that has a single codon for it. Everything else has at least 2. Some of them even have 4. If you look here at leucine, serine, proline, theanine, alanine, valine, these all have 4 codons for their particular amino acids. Here is basically an overview of DNA and transcription and translation. So if we take our DNA molecule, we have a blue one and a red one. And on this one, we're going to use the red one as our template strand. And so from here we have TAC, GAC, TGA, TGC. So now we are going to take and we're going to transcribe this. If we have T in DNA, we have an A in messenger RNA. An A in DNA, we have a U in messenger RNA. A C, we have a G, and if we have a G, we have a C. And so now we have made our piece of messenger RNA, and now from these codon sequences, we will have the particular tRNA that has a matching or complementary anticodon binding to the messenger RNA. Now, remember this, it is the codon sequence in messenger RNA that codes for the amino acid, not the anticodon. So AUG is the sequence for methionine, not UAC, okay? So as we go down, we'll see that we have methionine. AUG is our codon for methionine, so that tRNA comes and joins. Then our next codon is CUG. That's the code for leucine, so the tRNA with the corresponding anticodon comes in and binds, and so on and so forth, until we have a sequence of amino acids. If you look here at our last two, you'll see that they are both the amino acid threonine, but each of these has a different codon and anticodon sequence. ACU and ACG both code for threonine. So this is an example of that redundant code. This is kind of a picture of protein synthesis and how it all begins. The first thing that's going to happen is you're going to have your messenger RNA is going to be surrounded by your ribosome. So once our messenger RNA is bound within the ribosome, we're going to have the entrance of our two first tRNA molecules. You're going to have the first one brought into the P site and the second one brought into the A site. Once those are in the P site and the A site, a peptide bond will form between the two. Once that bond is formed, the ribosome moves down one sequence that what was once in the P site is now in the T site, or the E site, because this tRNA is empty now. And we have the one that was once in the A site, as the ribosome moves down, is now in the P site, and a new tRNA with an amino acid comes into the A site. So as that ribosome shifts, what was once in the A site is now in the P site. Let me get my cursor back here. What was once in the A site is now in the P site, and this new third amino acid is going to come in. Once we have those in the P site and the A site, a peptide bond now is going to form between amino acid 2 and amino acid 3. When that happens, these two amino acids now will leave the tRNA that's here in the P site, and they will bond to the one in the A site. The ribosome is going to shift down again. What was once in the P site is now in the E site. What was once in the A site is now in the P site, and a new amino acid comes in. So now we have amino acid 4. This is going to continue happening. You're going to have this the amino acid that's attached to the tRNA in the A site is going to form a peptide bond with all of the chain that is on the tRNA in the P site, and now the P site will be empty and the ribosome will shift down, and this will continue until it reaches this stop sequence, this stop codon. The polypeptide chain will be released, the ribosome will separate, go back to the beginning and bind to the start codon again, or it will dissociate because protein synthesis is completed. You have to have the termination of protein synthesis, and this is going to be brought about when you hit one of these termination or stop codons. UAA, UAG, and UGA are the three nonsense codons. They do not code for any tRNA. They code for a stop. The ribosome recognizes these three sequences as a stop sequence, and they will then separate and break the bond between the final tRNA and the finished polypeptide. Once that bond is broken, that chain is released. You'll have some modification of those proteins before they are released. They will fold on themselves. They'll achieve that tertiary conformation, and they will then be modified. That start codon will be clipped off. You'll have some cofactors added, and then they will join other proteins to form quaternary levels of structure, or they will be proteins that work functionally on their own. But this is as far as we're going to go when we're talking about the modification of proteins. We're not going to get into the specifics of folding or anything like that. If you look here, here is a picture of the whole process. We have our DNA that opens up, and we have our RNA polymerase. This happens to be prokaryotic RNA polymerase, because you can see that we are going through transcription and translation at the same time. So we have our RNA polymerase producing our messenger RNA, which is going to give us our transcription. Our start codon is open, our ribosome attaches and begins translation. As this transcript gets longer, we can also have more and more ribosomes added to this, and we can be producing more and more copies of our protein off of a single transcript. If you look over here, you'll see that you have this strand of messenger RNA, and all of these really dark blobs here are ribosomes. So you have numerous ribosomes, and you can see the proteins that are coming off of this. So you can see why some of the pictures that we have, some of the drawings that we have look the way they do. Here you have a ribosome with a set of amino acids coming off of it. And the longer the messenger RNA, the more ribosomes are able to go and translate that and give you more protein. Most of what we've talked about has been prokaryotic transcription and translation. Eukaryotic transcription and translation are a bit the same, but there are some significant differences there. They do start with a codon that is also AUG, but it codes for a different type of methionine. There are some modifications and structural changes in methionine between the eukaryotic and the prokaryotic methionine. Eukaryotic mRNAs are going to code for just one protein at a time. The presence of DNA in the nucleus means that transcription takes place in the nucleus, while translation takes place in the cytoplasm where the ribosomes are. So they can't take place simultaneously. In prokaryotic cells, transcription and translation can happen at the same time. Messenger RNA in eukaryotic cells have to pass through those nuclear pores and be carried to the ribosome before translation can take place. Most eukaryotic genes don't exist in an uninterrupted series of triplets like prokaryotic cells do. Genes are going to be broken into codable and uncodable sequences called introns and exons. Introns are going to be sequences of bases that do not code for a protein. And exons are going to be coding sequences that translate into some sort of protein message. In transcription, the entire gene, both exons and introns, are going to be read, and they're going to make something called a pre-mRNA molecule. That mRNA molecule is going to be modified to protect it from either end so that it doesn't get broken on either end. Actually, adenosine molecules are going to be added to the front and the back of the pre-mRNA molecule so that it protects it as it leaves the nucleus. It also is going to help direct it from the nucleus to the ribosome. Once the transcript is taken out, the exons and intron junctions are going to be clipped apart by enzymes called endonucleases. They are going to snip the introns out, and the exons then will be joined end to end to make a messenger RNA that can be translated then into a protein. In humans, 98% of our DNA are introns. So we have lots and lots of junk and very little codable information. if we look here, you'll see here you have your exon, here you have your intron. This is your DNA template. Here's our primary messenger RNA, and we're going to have enzymes called spliceosomes that are going to come in and bind to the exon portion. It's going to loop it up and make something called a lariat. And that enzyme then is going to splice out, these endonucleases are going to splice out these intron sections of the DNA, and then all of the spliceosomes, all of those enzymes are going to be released. The lariat portions are going to be excised, and these will be basically broken down and the nucleotides put in your cellular pantry, and all the exon portions will be spliced together, and now we have a messenger RNA that can be taken out of the nucleus and into the cytoplasm for translation. All of this modification from transcription, from the initial transcription to the splicing of the exons and introns out of the transcript, happens in the nucleus. Once that has all been clipped out, and now you have just exon messages here, that mRNA can go to the cytoplasm, to the ribosome, to be translated. Okay, that's the end of transcription and translation. Please, please, please take some time to write down any questions that you have or anything like that. The last portion of this is going to be regulation of protein synthesis and gene exchange, mutation, transcription, or transformation, transduction, and conjugation. So we're going to have probably another part of this that's going to be about the same length of material, but all of this should get us through to the end of the study guide, and you should be able to understand the process of DNA replication, transcription, and translation. I will take some time the next time we're together to draw out any of this that might be a bit confusing. I am also going to put in an email any animations that are going to be available to you. Those animations will probably come bundled together, and you should just be able to click on them and open them up, and they should work for you. If they don't, let me know and I'll fix them and send them back to you. So keep studying. Please read your chapter. Please go through these lectures and write down any questions that you have.